Okay, so we're going to start digging in and actually talking about some of these groups, and there's going to be a lot of names, so I'm going to try to keep these videos short so that they're not too overwhelming. So we're first going to start by talking about the first of the four supergroups, and the first supergroup we're going to talk about is a supergroup we call SAR. This is also going to be our biggest one. So this is a very large, very diverse clade. They're put together because DNA evidence suggests that they are monophyletic. The name SAR is just the first letter of the three subgroups within it, the Straminopoles, the Alveolates, and the Rhizarians. And one kind of interesting thing we see in this group is that some members have undergone secondary endosymbiosis, and some members have undergone both secondary and tertiary endosymbiosis. So we're going to talk about these three subgroups within this supergroup, starting with Straminopoles. So within the Straminopoles, there are actually three clades that we'll talk about: the Diatoms, the brown algae, and a group called the Umacetes or Umacetes. I never can decide how I want to say that. Which are known as the water molds. But they're not really mold. So there's a lot of variety here in terms of nutritional category. We have photosynthesizers. So some do photosynthesis, some are heterotrophs that have to consume organic matter from another organism. One thing that we see that unites most members of this subgroup is the presence of a flagellum with hair-like projections. And in not all but many of them, there's also a second shorter smooth flagella, say plus. So I should say that this flagella is more apparent in some organisms than others in this group. So the name Straminopoli, that stramin comes from the word, the root word for straw. So kind of this, kind of referring to these short hair-like projections we see on one of those flagella. So let's go ahead and start talking about these subgroups a little. And yes, this is going to be a lot of taxonomy here. So let's talk about the diatoms. These are unicellular algae. So again, some protists are multicellular, some are unicellular, most are actually unicellular, and this is one of the unicellular groups. They have shells that are made of two sides that kind of overlap like a petri dish, and on some of these, maybe this is more apparent on some of the shells than others. I know it's hard to see my mouse, but this one that's kind of got a triangular shape, you can kind of see kind of how the two halves fit together like a petri dish. Let's pluralize that correctly. If you don't know what a petri dish looks like, let me know. I'll show you one in lab. They are a major component of this shell is silica. So silica embedded within kind of the organic matrix. So silica is a major component of sand and is also used in making glass. So they have like a very hard, sort of glass-like shell. With really intricate and beautiful designs. These are one of the most beautiful organisms that we'll look at all semester. We'll be looking at some of these under the light microscope. And they're very important ecologically because diatoms are a major component of the phytoplankton. So in marine ecosystems, there's what we have what's called zooplankton, which is our little small animals, things that like maybe baleen whales would be eaten. But then there's also the phytoplankton, which are extremely small organisms like these diatoms that are important photosynthesizers, creating, so they do photosynthesis, which of course leads to O2 production. So these are very important base of food chain in marine ecosystems, and also very critically important in global oxygen production. So these are a really ecologically important group. Okay, so still supergroup SAR, still subgroup Straminopolis. We have our brown algae. So our brown algae are the largest by far, largest and most complex algae. They are all multicellular. And in fact can be enormous. This would include things like the giant kelps that make up those giant kelp forests you might have seen in images on nature shows or something. They have structures that are analogous to plant structures. You can see them in this image, what's called the holdfast, the stipe, and the stem. I'm sorry, the holdfast, the stipe, and the blade. So the holdfast are superficially, they kind of look like roots, but they're not roots. They don't have the same internal structure of a plant root. They don't pull water up from the ground, right? They wouldn't need to. These organisms are marine. But what they do is they play an anchoring role, helping anchor this organism to whatever substrate, the rock usually, so that they don't just kind of float away. The stipe is analogous to a plant stem. It's sort of the main axis of the organism and will sort of lift the blades up further so they can intercept more sunlight, because those blades are analogous in function to a plant's leaves. They're flattened so that they can intercept more sunlight to do more photosynthesis and play the role of the main photosynthetic organ of the organism. So superficially, but not homologous to plant structures. And now let's talk about the water molds. And they get called molds because they have some things about them that sort of are superficially similar to fungi, but they are not fungi. They are a protist. So they're filamentous. So in that way, they kind of look superficially like a fungus. They're filamentous. They're stationary. Okay, these things don't move around. They are absorptive heterotrophs. Which means they have to get their energy from another organism, and they absorb it. So what they do is they secrete enzymes onto and into whatever it is they want to eat, and allow those enzymes to digest and break down that tissue outside of their body, and then they absorb the nutrients after they're broken down. And once they're broken down into smaller molecules. We would call these saprobes. I can't spell this morning. There we go. I'll get my B and my O in the right order. Saprobes are organisms that feed on dead organic matter. So they are decomposers. So they play an important role ecologically as decomposers. Now, like I said, their growth is analogous and has some similarities to fungi, but one of the things that's not like fungi is that they have a cell wall made of cellulose, whereas fungi have a cell wall made of a structure called chitin, and we'll talk a lot about that when we get to the fungi. Okay, so that is our water molds. So the last subgroup that we're going to talk about in the SAR, sorry, no, this isn't the last, this is the second. I'm sorry, I'm ahead of myself. The subgroup Alveolata. So members of this subgroup get their name from membrane-bound sacs called alveoli that are found under their plasma membrane. And the function of these alveoli is not completely understood, but there are some ideas that it may be important in supporting the cell surface. There's some other hypotheses out there. And this picture, this micrograph here, shows these little sacs, these alveoli. In this case, we see them on either side of the flagellum of this organism. So this is a group that is all unicellular. Most are going to be photosynthetic. They don't have room. Although there's going to be some exceptions. And there are three subgroups that we're going to talk about within, or sort of three clades within this subgroup we're going to talk about within the alveolata. We're going to talk about a group called the Dinoflagellates. We're going to talk about a group called the apicomplexans. And a group called the ciliates. So their names tell us a little something about us. The dinoflagellates have flagella. The apicomplexans, okay, the name doesn't really give this away, but the apicomplexans are a group of pretty much all parasites. And in the ciliates, they have cilia. Okay, here's a dinoflagellate. And I always, at first I thought that dino meant, oh, it's like a dinosaur. Well, dino actually comes from a word meaning to spin, because the way that this organism moves around, actually it has kind of a spinning motion as it moves. So, but I still like to think about the word dinosaur. Helps me remember. So let's talk about our dinoflagellates. Okay, so their cells are surrounded by cellulose plates. That make up kind of like an armor. And there are flagella. There's actually usually two flagella. There's one that we find in the groove between the plates. And then typically there is a second flagella trailing. So let's see, this image, yes, we have So one of the red arrows is showing what we call the flagellar groove, which is something you're going to look for in lab when you get these guys. If you don't see the flagellar groove under your microscope, it might mean you're not quite in crisp focus. And then we have two of these arrows kind of pointing to the one of the flagella kind of goes along the midline of the organism and lays along this flagellar groove. And then we have a second flagella that is kind of, I guess you would say, a terminal flagella coming out, one end of the organism. So I need more room, so I'm going to erase this stuff, but we're just going to remember our taxonomy here. So Dinoflagellate, they spin as they move, in part because of the way these flagella are set up, or maybe completely because of the way these flagella are set up. They are a huge component of the phytoplankton. So they have important roles as being part of the photosynthetic plankton, so important roles with photosynthesis, and as, being part of the base of the food chain. So they play an important role there. And some can be in what we call a symbiotic relationship, and which, remember, means we're living in close proximity or even inside another organism. With other organisms such as corals or other protists. For example, when we get to the point, when we get to animals and we're talking about corals, we're going to talk about some dinoflagellates that act as symbiotic partners with them. One of the things that dinoflagellates are responsible for is a phenomenon known as red tide, which you may have heard of. Explosive population growth of dinoflagellates can lead to something called red tide. So the water here can cause what we call red tide, and the red comes from the fact that the carotenoids, which is the type of photosynthetic pigment in many of these dinoflagellates, appears red or pink. And so if you get a very large accumulation of them at one time, you can actually make the water appear red. So these are unicellular, so think about how many must be in this image for the water to actually have that red streak like that. Some species produce toxins that can actually cause things like massive die-offs of fish and other invertebrates, and also be toxic to humans. So they can actually be very problematic, and every once in a while you'll hear a warning, don't go in the water, or don't eat the shellfish because of concerns with the toxicity of red tide. Okay, so another group that can be a little bit of a problem to humans is a group called the apicomplexans. These are all animal parasites. And some can cause human disease, such as the Plasmodium we're seeing in this image. This causes the disease malaria. So they get their name in part because at one end of the cell, what we would call the apex of the cell, there is a complex of organelles that are specialized for penetrating host cells. They typically have a complex life cycle that includes both sexual and asexual phases, and often involves multiple hosts, which is what you're looking at here in this image. So Plasmodium actually has two different hosts, humans and mosquitoes, which is why when we talk about concerns about malaria, we're typically, we approach that by trying to control the mosquito population. So this Plasmodium organism, in its life cycle, and it's a cycle, so I guess you can start wherever you want, but we'll say, one form of the organism exists in the mosquito, and then when the mosquito bites the organism, bites the human, or, and it can transmit the Plasmodium into the human, where it can then grow and develop and reproduce and produce gametes, and then when the mosquito gets, that the human host gets bit again, those gametes get released back into the mosquito, where they fuse, and we start the cycle all over again. So it's kind of multi-host. Sorry, that's my. Oh, sorry. Sort of this multi-host, multi-form organism. So it's really complex. We're going to look at some apicomplexans in lab, and we'll talk a little bit more in detail about, kind of some of the stages of the life cycle and what we want to be looking for when we look for this organism. Okay, so finishing off this subgroup, of Alveolata are the ciliates. So the ciliates are a very large and varied group. They get named for their mode of movement and feeding, which are cilia. Cilia are short, hair-like structures that are typically found in very large numbers, covering typically the whole organism. This is a really interesting group because they have two types of nuclei. A one or more tiny micronuclei, and a larger macronucleus. And these two types of nuclei are going to be kind of the key to genetic recombination in this organism. Because this organism, in this group of organisms, we have genetic recombination via conjugation. Now that name might sound familiar from the bacteria lecture. All right, that's where two organisms within the same generation are able to swap genetic material. So these ciliates, what we'll see is that they actually divide themselves by a process similar to binary fission, where they're just going to split. But they get their genetic recombination through this process of conjugation. And later we're going to talk about sexual life cycles at the end of this lecture, and we'll get into the details of how this works. So almost all ciliates are heterotrophic. A few do possess photosynthetic capability, usually through some kind of endosymbiont. So yes, and we'll be looking at ciliates in lab as well, and you should actually be able to get an image much like the one, my microscope image you see here under your light microscope. It'll, it has to be in really good focus, but it's doable. Okay, so I'm going to stop this lecture here because it's getting a little long, and we're going to pick up with the subgroup Rhizaria when we come back. So see you soon.